Hello everyone, I am Manoranjan Burman and I welcome you to my YouTube channel. Today I am going to discuss about a deadly fungal disease which is called as mucormycosis. Nowadays it is called as black fungus by the general public, which is totally wrong. The infected lesions turns into black color. Maybe because of that the disease is being called as black fungus, but actually not. Mucormycosis is an invasive fungal disease caused by the group of Mucormycetes by the species of Rhizopus, Mucor and Absidia. It is an opportunistic fungi which progress rapidly and it is highly destructive. The fungal disease may be fatal in 7 to 10 days if not treated early. Then we will have introduction of mycology. Then we will see the habitat and mode of infection of the disease, risk factors, which group of individuals can get the disease. Then we will have a look at the pathogenesis of the disease, how the disease progress in the human being. Then we will check the clinical manifestations of the disease. Then we will have clinical features, what are the signs and symptoms depending upon the site involved. Finally, we will have a discussion on the laboratory diagnosis, how we can diagnose this type of fungal disease in the laboratory. Before going through the mucormycosis, let's go through the basics of mycology so that you can have better understanding of mucormycosis. You know that microbiology has different branches like bacteriology, where we study about bacteria, virology, where we study about different types of viruses, mycology, where we study about the fungus. Most fungi has beneficial effect only because they reside in nature and they break down and recycle organic matter. Some fungi contribute to the production of food and wine also. Some fungi provide some useful bioactive secondary metabolite known as antibiotics. On the other hand, there are only a few hundred species of fungi which have been implicated in human disease. Fungi grow in two basic forms, yeast and molds. Yeast is the simplest form of fungi which has a round or oval cell. Some fungi forms elongated tube-like structures known as hyphae. Hyphae may be septated and non-septated. Septated hyphae has cross wall in between the cell, but the non-septated hyphae do not have cross wall in between the cell. That is an important point. Mucormycetes produces a non-septated hyphae, which we will study today. A tangled mass of hyphae are known as mycelium, and the fungi that can form mycelia are known as molds or filamentous fungi. We can classify fungi based on their morphology as yeast-like fungi, molds or filamentous fungi, then dimorphic fungi. Yeast are round or oval unicellular fungi. They reproduce by an asexual process called budding. Yeast forms smooth and creamy colonies on a culture media. Cryptococcus neoformans is an example of pathogenic yeast which can cause disease in the human being. Yeast-like fungi grows partly as yeast and partly as elongated cells resembling hyphae. Yeast-like fungi can form pseudomycelium. Pseudomycelium means it is not actually a mycelium. Pseudo means false. It is a false mycelium. Actually, it is not a mycelium but looks like mycelium. You can see this structure. It looks like mycelium but actually it is not a mycelium. Candida albicans is a pathogenic yeast-like fungi. If I say pathogenic means it will be able to cause infection in the human being. Mold or filamentous fungi. This forms true mycelia or true hyphae and reproduce by the formation of different spores. Hyphae are delicate structures which can be destroyed easily by harsh environment conditions like heat, cold, etc. But the spores are resistant to harsh environment conditions. In harsh environment conditions like heat and cold, it can survive well. The examples of molds are Aspergillus, Mucor, Rhizopus and Absidia. Then at last the dimorphic fungi. Dimorphic means double morphology. They will have two morphology. So you can remember they can form two forms, mold and yeast. So mold in the cold and yeast in the hot. The example of dimorphic fungi are Histoplasma capsulatum. Fungi is divided into four groups based on the production of sexual spores as zygomycetes or mucormycetes. These fungi have non-septate hyphae. It forms asexual spores called sporangiospores which contains within a sporangium. The sexual spores are known as zygospores or oospores. Next is ascomycetes. Ascomycetes has septated hyphae. It forms sexual spores known as ascospores. The spores will be within a sac or ascus. Next is basidiomycetes. Basidiomycetes are septated hyphae. It forms asexual spores called basidiospores. The basidiospores are on a basidium or a base. The last one is deuteromycetes or fungi imperfecti. Deuteromycetes are septated hyphae. Its sexual state is not clearly known. The infections caused by fungus are known as mycosis. We can classify fungal disease as superficial mycosis. These infections are limited to the outermost layer of the skin and hair. The example is Tinea nigra, white piedra, black piedra. Next is cutaneous mycosis. These infections that extend deeper into the epidermis as well as invasive hair and nail disease. Dermatophytes are an example of cutaneous mycosis. Next we will talk about subcutaneous mycosis. This infection involved in the dermis and subcutaneous tissue and underlying muscle and organs. Chromoblastomycosis is an example of subcutaneous mycosis. Next we will talk about systemic mycosis. These infections will originate primarily in the lungs but may spread to other organ systems. Blastomycosis is an example of systemic mycosis. The last one is oppurtunistic mycosis. As the name indicated, the infections are not harmful in healthy individuals, but it can cause infections to patients with weak immune systems like AIDS, cancer patients, uncontrolled diabetes mellitus, taking steroids. Aspergillosis, zygomycosis and mucormycosis are example of opportunistic mycosis. Mucormycosis is the one which we are going to discuss now. Nowadays, some patients after recovering from COVID-19 is getting this mucormycosis. Remember, not all the COVID recovered patients are developing mucormycosis. Mainly the COVID infected patients with uncontrolled diabetes mellitus or diabetic ketoacidosis or those who have taken steroids for their treatment. Now let's talk about the habitat of mucormycosis. That means where does it live? It can live in soil, decaying vegetative matter, decaying fruits, drying leaves, compost. You can have a look at the picture. Now the question is, how do they enter from these places into the human body? I am talking about the mode of infection. Inhalation of the spores. During respiration, you can inhale the dust along with the spores through air. Ingestion of spores, eating food contaminated with these spores. Direct contact of spores to the injured skin. Now I am going to talk about the risk factors. Risk factor means who can develop the disease. Persons having uncontrolled diabetes mellitus or diabetic ketoacidosis can develop this disease because these persons will have impaired phagocytic functions. Their macrophages and polymorph nuclear cells are dysfunctional. They cannot phagocytose the spores. As a result, they will develop the disease. Then person with iron overload or iron chelators. The iron chelators will make the iron free and free iron will be taken up by the fungi for their growth and development. Person with corticosteroids. We know that corticosteroids are immunosuppressor. Immunosuppressors can lower the level of immunity. As a result, they are prone to get infections easily. Organ or bone marrow transplantation. To avoid rejections of the transplanted organ, immunosuppressors are used. So as a result, the immunity level will be low and they will easily prone to get the disease. Next is hematological malignancies. In the case of hematological malignancies, there will be suppression of immunity. The person may go for chemotherapy. As a result, there will be decreased neutrophils. There will be neutropenia. Person with neutropenia can develop mucormycosis. Next is COVID-19 in diabetics. After recovering from COVID-19, some patients with diabetics develop mucormycosis. In case of diabetic patients, the diabetic decreases the immunity. And in some cases, patient is treated with steroid. As a result, there will be hyperglycemia, increased level of blood sugar. Then the diabetic will be uncontrolled, the sugar will be uncontrolled. In that case, the persons can get the disease easily. Next is trauma and burn. Trauma and burn can cause cutaneous mycosis. Next, we will have a look at the pathogenesis, means how the disease involves. The fungal spores enters via nose. After entering through the nose, in case of healthy individual, the macrophages and polymorphonuclear cells will engulf the fungal spores and destroy it. Engulf means it will eat up the spores. So in the case of healthy individual, the disease will not progress. In the case of high risk individuals, the macrophages and the polymorph nuclear cells cannot phagocytose the fungal spores because of defective chemotaxis and phagocytosis of the immune cells. First, the nose is affected. As it is a destructive fungi, it will destroy the cribriform plate and reach the brain. Via nose, it can reach the orbit, means it can reach the eye, it can reach the paranasal sinuses. Via nose, it can destroy the palate and it can reach the lungs also. If the fungus is getting entry by ingestion, then the stomach is involved and reaches the intestine. Intestinal involvement are rare. If the fungus contact with injured skin, then the skin is involved. In case of healthy individuals, the fungi cannot infect. But in case of high risk individuals, the spores will germinate into the hyphal form. The spores will become hypha in the body. The spores are resistant from the environment. Spores will not get damaged. They get entry into the body and they will become the hyphal form. The hyphal form causes inflammation in the body. So the inflammation will result in the clinical manifestations. The mucormycosis is angioinvasive. That means it can invade the blood vessels. Once it will invade the blood vessels and entry into the blood vessels, then it will cause thrombosis. Thrombosis means formation of solid mass. Thrombosis means formation of solid mass in the blood vessels. As a result, it will cause infraction. Infraction leads to ischemia. Ischemia means inadequate blood supply to a certain tissue. If the blood vessel is blocked because of thrombosis, those tissue will not get sufficient blood supply. As a result, the tissue will suffer from hypoxia. Hypoxia means lack of oxygen. So hypoxia will result to necrosis. Necrosis means tissue dead. The tissue will die because of lack of oxygen. The dead tissue will form black crust. So this is the characteristics, clinical manifestations of mucormycosis. So it will form black crust. Now we will move on to the clinical manifestations. To know the clinical manifestations, we need to know the types of mucormycosis. Based on the site involved, there are many types of mucormycosis. Rhinocerebral mucormycosis. The rhino means nose, cerebro means brain. That means it will involve in the nose and brain. Rhino-orbitocerebral mycosis. Rhino means nose, orbit means eyes, and cerebral means brain. It will involve in the nose, eye and brain. Then pulmonary mucormycosis. Pulmonary mucormycosis will be involved in the lung. Then gastrointestinal mucormycosis. It involves in the stomach and intestine, but intestinal involvement is rare. This is cutaneous mycosis. Cutaneous mycosis involves in the skin. Next is disseminated mucormycosis. In the disseminated mucormycosis, the fungi reach the blood. Once it will reach the blood, through the blood circulation it can reach to any organ. It can infect any organ. So this is the last stage of the disease. Next we will discuss about the clinical features. If nose is involved, then nasal congestion will be there, nasal blockage will be there, and you can see black crust in the nose. In the nasal secretions also you can see some black particles. In case of paranasal sinuses, there will be paranasal pain, paranasal swelling and paresthesia. In case of palate, you can see black crust in palate. So these black crust are the common and important signs. If it involves in brain, then there will be headache, vomiting, seizures. It affect the orbit or eye. There will be diplopia, ptosis and periorbital cellulitis. Diplopia means person may see two images of a single object. Ptosis means dropping or falling off upper eyelid. And periorbital cellulitis means there will be redness swelling area surrounding the eye. Eventually there will be abrupt vision loss. The person may lost his vision. The mucormycosis can damage nerves also. Paranasal pain is associated with fifth cranial nerve damage, that is the trigeminal nerve. Person with diplopia and ptosis is associated with third cranial nerve damage, that is the oculomotor nerve. If it infects the lung, there will be chest pain, hemoptysis and dysphagia. Hemoptysis means blood is in sputum, dysphagia means difficulty in breathing. If it involves in stomach and intestine, there will be ulcer and hemorrhage in the gastrointestinal tract. If it infects the skin, then there will be papules and pustules. Papules means raised area of skin tissue that is less than 1 centimeter around. Pustules means small bumpy area of skin contains pus. From the pus, the fungi will grow and it is known as hairy pus. Next we will discuss about the diagnosis of mucormycosis. Early diagnosis and early treatment leads to good prognosis. There is more chance of survival. It is a rapid and destructive fungi, so delayed diagnosis and delayed treatment leads to poor prognosis. As a result, there is less chance of survival of the patient. If untreated, the patient may die within 7 to 10 days. Now let's talk about sample collection. Tissue biopsy is the best sample. Or you can take deep nasal swab, nasal secretions also. Remember, do not homogenize the biopsy sample. It will damage the hyphae, resulting no growth in the culture media. After collection of the sample, we need to examine the sample under the microscope. That is sample microscopy. In the sample microscopy, first we will perform KOH mount. We use 2-3 drops of potassium hydroxide. Potassium hydroxide will digest the cell and tissue, proteinaceous material, and so that the fungal hypha will be clearly seen under the microscope. Then you can perform histopathological staining, that is Grocott, Gomori, methenamine, silver, GMS in short. Then we can use fluorescent staining, that is chalcofluor white stain. Chalcofluor enhances the detection of fungi because chalcofluor white will bind with the cell wall of the fungi. After preparing the sample microscopy, once you will observe the sample under the microscope, how will you identify the fungi? Key point to identify mucormycosis. They have broad hyphae comparing other fungi. It can be 8 to 16 micrometer. They do not have septum unlike other fungi. They are aseptate. They form right angle branching hyphae. After sample microscopy, we need to prepare culture media. and inoculate the specimen into the culture media for their isolation and growth. The commonest culture media is SDA media, sober dextrose agar. Mucormycosis shows both vegetative growth and aerial growth. Vegetative growth means the fungi will grow on the surface of the culture medium. Aerial growth means the fungi will grow above the surface of the culture medium. Because of the aerial growth, it looks like cotton candy. The fungi can grow above the surface and lift the lid. So it is known as lid lifter also. If you look at this picture, you can see the aerial growth of the fungi. Initially the fungal colonies are white and later they can become greyish in color. But the Rhizopus is an exception. Rhizopus will not have white colonies. Initially the Rhizopus will show white growth with black dots. Can you see in this picture? This growth is known as salt and pepper appearance. The growth of Rhizopus is known as salt and pepper appearance. Have a look at this picture. Then we are going to check the culture microscopy. We need to take some fungal colony on a slide and stain it with lactophenol cotton blue, that is LPCB stain. Under the microscope you can see broad non-septate right angle branching hyphae. This is the unique characteristics of Mucormycosis. On culture medium, Mucormycosis produces both hyphae and spores, but in sample microscopy we can see only hyphae, not the spores. So if we take fungal colony from a culture medium and observe under the microscope, we can see hyphae as well as spores. Mucormycosis has two types of spores. Sexual spores, which are known as zygospore or oospore. Asexual spores, which are known as sporangiospores. Sporangiospores are formed within a sporangium. This is the sporangium. Inside this sporangium, sporangiospores are there. The sporangium is developed on the end of the hyphae called sporangiophore. This is the sporangiophore. This is sporangium. Inside the sporangium, sporangiospores are there. 3 points you need to remember: sporangiospores, sporangium, and sporangiophore. Remember, sexual spores are not produced in the SDA media. It is produced only some special media. Rhizopus, Apsidia and Mucor, this causes mucormycosis. This can form broad non-septate hyphae. It can form right angle hyphae. Now you may think how to identify rhizoid, Apsidia and Mucor under the microscope. How to differentiate Mucor, Rhizopus and Apsidia? Have a look at this. This is a non-septate hyphae. It forms right angle hyphae, and at the end of end hyphae there is a sporangium. The sporangium contains the spores, and the end hyphae which contains the sporangium is known as sporangiophore. And there is an important point. This is called nodal point. From the nodal point, the sporangiophore develops, and these are known as rhizoid. So for identifying the Mucor, Rhizopus and Apsidia, the non-septate hyphae, sporangiophore, sporangium, all the same, but the difference is the rhizoid, the position of the rhizoid. So this is the important point. This is a picture of Apsidia. Now have a look at the Rhizopus and Apsidia. See the difference. The Mucor will not have rhizoid. Others are same. It has non-septate hyphae. It forms right angle hyphae. It got sporangiospores, sporangiophore. In case of Rhizopus, there is rhizoid. Apsidia also there is rhizoid. Now you see the difference of Rhizopus and Apsidia. The Rhizopus and Apsidia both has rhizoids, but the pattern of the rhizoid is different. In case of Rhizopus, the rhizoids are developing just below the nodal point. In case of Apsidia, the rhizoids are not developed just below the nodal point. So it is called internodal rhizoids. This is called, in case of Rhizopus, the rhizoids are known as nodal rhizopus. In case of Apsidia, the rhizoids are known as internodal rhizoids. Rhizoids are the only difference to identify the Mucormycosis and Apsidia. Everything is same except the rhizoid. Next technique is polymerase chain reaction, in short PCR. By using PCR, we can detect the specific gene of this fungi and make the diagnosis. CT MRI can also be done based on the site of involvement, and it can give information of the extent of the spread of disease. This is all for today. Please like, share and comment. And those who are new to my channel, please subscribe to my channel. Thank you for watching. Thank you.